In the 1990s, Roberta J. Pentneri, professor of anatomy and cell biology at the University at Buffalo, and her colleague discovered the chronic alcohol use did not cause nerve cell death. In fact, their conclusions were that alcohol causes 1. Changes in the shape of neurons. 2. Altered connections between neurons. These effects are partially reversible with discontinuation of alcohol. What parts of the neurons are affected by alcohol? The parts of the neurons affected by alcohol are called dendritic spines. What are dendritic spines? Neurons consist of incoming fibers, or dendrites, which are shaped like a tree. Dendron is Greek for tree. The dendrites have spines rather like leaves on a tree, which contain synapses which connect to surrounding neurons. Synapse means connection in Greek. The synapses on dendritic spines receive information from other neurons. Each neuron passes information onto other neurons by a single fiber called the axon. Axon means to go or to travel in Greek. Why are dendritic spines important? Dendritic spine numbers and shapes are very important in psychiatric disease. Most psychiatric diseases are associated with abnormalities of the shapes and numbers of dendritic spines. How do changes in the shapes and numbers of dendritic spines affect thoughts and feelings? When dendritic spines are less dense and shorter in the front of the brain, known as the frontal lobes, the bad feelings of excessive self-pity, fear, resentment, and dishonesty predominate. These changes are often associated with obsessive-compulsive behavior. When dendritic spines are more dense and taller in the frontal lobes, the good feelings of gratitude, acceptance, trust, and honesty predominate. What is the effect of alcohol on dendritic spines? Further studies by Zhu et al. Have confirmed that alcohol causes altered density of dendritic spines and altered size of dendritic spines. These are described as reactive dendrites, or more conveniently dendritis. What is the effect of having greater or fewer dendritic spines on electrical firing rates? There appears to be a causal relationship between dendritic structure and firing rates. The more dendritic spines there are, the greater the firing rates of neurons and good feelings. The fewer dendritic spines result in lower firing rates and bad feelings. Altered dendritic firing rates have been noted with alcohol exposure. Occasional use increases dendritic spine lengths and results in reduction in fear or Dutch courage. Chronic use of alcohol results in reduced dendritic spine lengths and numbers and bad feelings. How does alcohol cause changes in dendritic spines? Alcohol is a prime example of a neurological immunogen. There is increasing evidence that the immune system, and in particular regulatory T cells, macrophages, and dendritic cells, are important in controlling central nervous system inflammation. The activated immune system results in substances called cytokines being released, which cause inflammation. There is also increasing evidence that there are many abnormalities of the cytokines of the immune system in other psychiatric diseases, for instance depression and schizophrenia. Other obsessive-compulsive disorders are known to be associated with immunological abnormalities, for instance following infection. Why are some people affected and not others? The genetic makeup of the patient is important. Often there is a family history of alcohol dependency. Genetic predisposition has also been noted in most psychiatric diseases. Psychiatric diseases are more prevalent in alcohol dependency, for instance depression, anxiety, bipolar disease, and schizophrenia. What other changes are there in the nervous system in alcohol dependency? There are many neurotransmitter, receptor, and biochemical abnormalities well documented in alcohol dependency. These may contribute to the inflammation. How could increasing dendritic firing rates help? Further evidence of the importance of dendrite firing rates in psychiatric disease was confirmed when gene therapy was used to increase dendrite firing rates and led to the improvement in the autistic clinical behavior, such as self-obsession in mice with Fragile X syndrome. How could reducing immune system activity help? When cytokines were reduced in Alzheimer's disease using a cytokine blocker, there was an improvement in memory within hours. In summary, alcohol and drug dependency, or alcohol-drug dendritis, is due to chronic alcohol use resulting in an abnormal immune response in genetically predisposed people, resulting in reduction in dendritic spine numbers. Shorter and more sparse dendritic spines are associated with reduced firing rates and the thoughts and feelings of resentment, fear, self-pity, and dishonesty. There is an associated obsession-compulsion to drink more alcohol. Occasional alcohol use results in taller and increased numbers of dendritic spines. Taller and more dense dendritic spines are associated with higher firing rates and the good feelings of gratitude, acceptance, trust, and honesty. This is described as conviviality. Abstaining from alcohol results in the dendritic spines regrowing and reconnecting with other neurons. This is associated with the development of good feelings and a reduction in the obsession-compulsion to drink alcohol. Perhaps the future of treatment of alcohol and drug dependency also lies in the field of immunology, as with many other psychiatric diseases.